Nathalia will be talking. She's been a PhD student with the corn breeding program for last 3 years or so. She recently took a job with Bayer Crop Science. Actually, she's working for Bayer as we speak, but living here in College Station for another week longer. Nathalia is from Brazil and did her undergraduate in Brazil, but she actually got a master's in France in remote sensing before coming here to join our program. And she was one of 2 students. I believe the other one is Jales, who was funded by a special Monsanto program. It was Monsanto at that time to bring in Brazilian scholars and get them PhDs. So I hope I didn't miss anything, Nathalia. I'm sure you're going to talk about your exciting research. And again, I think she's done stuff that nobody in the world has really done to this point. And I was pleased to hear her tell us the other day that our data set is more robust temporally than some of the data sets that she's gotten to work with so far in her new company job. So exciting stuff ahead. So without further ado, thanks, Nathalia.
Thank you, Dr. Murray. And thanks to everyone for being here. Well, today I'll be talking a little about one of my research projects that I've done during my PhD. From UAVs to yield modeling, the use of phenomics in a maize breeding program. Just an overview of this talk. First, I'll give you a little background on this project, the use of phenomics in plant breeding. Then I'll be talking about phenotyping with maize for grain yield prediction. I'll talk a lot about the phenotyping with UAS, genetic variance components, and also yield prediction modeling. The trials that I used for this project were part of the Genomes to Field initiative. Genomes to Field initiative is an extensive network of genome by environment trials. It started in 2013, supported by the Iowa Corn Promotion Board. And until 2019, over 180,000 plots have been evaluated in 162 environments in North America, and more recently, some environments in Canada and Germany have been added. So basically, for each trial, the PIs are responsible for collecting some core traits, and also there is weather station collecting data. But the PIs can also add additional phenotypes, and that's what we did here for College Station. We started using UAVs to collect data. And I think it's never, it's always good to reinforce that plant breeding is this cycle of creating genetic variation, selecting the best genotypes, conduct replicated testings, and releasing improved cultivars. And if we think as a phenotype as a result of the interaction of the genotype, area, and environment, it has to be phenotyping so we can generate gains like in plant breeding. So the progress in plant breeding is a function of this, mainly these 3 items: the length of the cycle of the plant, and by that I mean if in 1 year I can have more cycles, so I'll be able to get more genetic gain in the same amount of time. It also is a function of the number of progenies evaluated. So if you can evaluate more individuals, so you'll be also making more decisions. But it's also important to make accurate evaluations so you're not moving into the wrong direction. So phenotyping and high throughput phenotyping is it's able to act on these 2 last items. So if you phenotype with UAVs, for example, you are able to evaluate a higher number of progenies because you are able to acquire data much faster in a much extensive area, but also you will have increased accuracy and evaluation as some bias will be removed. And I will talk about that on the next slide. So phenotyping is a bottleneck in plant breeding. So usually how phenotyping has been done, it was manually, and it's a synonym of laborious, time-consuming, tedious job with the potential increased source of error. Because imagine, like, if you have multiple people collecting data on the field, they're not collecting data in the same way, and even if the same person collects the data, the performance is not the same from the beginning of the day to the end of the day when you're exposed to heat and to bugs. There's also a limited number of measurable traits that you can do manually. And if we talk about like collecting data with combines, you have to travel long distances with heavy machinery. So if you have an UAV, you could do that much faster because of very light equipment. The UAS-based phenotyping, it reflects into a reduced workforce and a faster area coverage. It has capability of phenotyping more plants in the field and with a high spatial and temporal resolution, and also a broad set of phenotypes. If you have in mind that you can mount into a UAV, different sensors, so multispectral, thermal, RGB cameras, and even LiDAR. So with that background set, the hypothesis of this study is that the remote sensing derived phenotypic variables that we can acquire from UAV imageries, those variables, they are heritable measurements that can predict yield. So that's our hypothesis here. And the objectives of this study was first, develop a data extraction pipeline so we could know each step to extracting, to going from unstructured data, that is our images, to structured data. So we could go from the images to a table with your plot IDs and then the variables that you want to study. So once we have all our data structured, we're going to explore the variability of each variable and see which proportion of that variability can be explained by a genetic component. Third, we also want to look into how useful are those variables for predicting maize grain yield, and also to determine which critical moments to go in the field and collect data with UAVs. The field experiment used in this study was conducted in 2017 in College Station, Texas. We studied 250 hybrid maize genotypes under 3 treatments. One treatment is optimal planting date in dry land, so we could create this water stress. The second one was optimal landing date and irrigated. And the third one was late planting, so we could have a heat stress and also irrigated. Each of the 250 hybrids was replicated twice in 2-row plots. Something important to point out, even though we had 3 treatments, for the sake of this study, we're looking into only 2. And the reason why is because we're dealing with a time series, and then we're not able to compare these 2 treatments with the one that was planted later. So for the studies, we're dealing with optimal planting date dry land, and then optimal planting date irrigated. The unoccupied area system that we used was a fixed wing tough wing UAV with a Sony high resolution RGB camera a flight altitude with 120 meters with a flight overlap of about 75 to 80%, which gave us a mean spatial resolution of 2.7 centimeters. That means that one pixel in the image would cover 2.7, like a square in the ground with 2.7 centimeters. The data collection, so the phenotyping periodicity. The flights were done weekly throughout the growing season, and during the flowering period, we flew twice a week. We were interested in three main phenotyping categories, phenotype categories. Number one being plant height, the other one being vegetation indices and canopy cover. And for the G2F project, we also collected manual data. So we collected flag leaf and tassel heights, and also flowering notes on days to silk and days to anthesis. And these are all the steps it took us to going from the UAS survey until yield modeling. So it's a very nice concept when you talk about it. It may sound easy, but it takes a lot of steps until we get to the results. So step number one would be going to the field, placing ground control points. And then, so every week, collecting data with UAS. Once we had the images were mosaicked, so they were stitched together, and also they were ortho-ratified. That means they were corrected geometrically. With that, with the images, we were able to produce vegetation indices. And with a technique called structure from motion, it was possible to recreate the plants in 3D from a point cloud and get a canopy height model. We also identified the plots and computed the canopy cover, which is a ratio between the amount of pixels in a plot and the amount of pixels that are covered by vegetation that represent vegetation. The data was extracted by plot with data initial statistical analysis, looked into the genotypic values estimation, repeatability of each variable, and then we selected the variables for yield modeling. Those are the main steps. A little bit more of details on the phenotypic variables we were studying. So we'll look into three different vegetation indices. Number one being normalized green-red difference index. So all of them are with RGB bands. So NGRDI is the equivalent to NDVI, but instead of using the infrared band, we use the green band. Axis green and axis green and axis red difference. Those three are widely used for like agronomic purposes. Canopy cover. So canopy cover, first we do a binary segmentation of plant and ground. So we're able to tell from each pixel what does that pixel represents, if it represents vegetation or ground. And also the canopy cover enables the VI extraction from vegetation pixels only. So there are two ways of dealing with the vegetation indices. So one is getting, for example, a mean value over the entire plot. And the other one is to removing the ground and looking into the values coming from the plants only. And finally, plant height. And the step to getting plant height was getting the point clouds and then computing the canopy height models. And then we extracted for each plot. We got the 9/5 percentile of plant height. And then we have it extended because we can, from those variables, we can combine them in different ways and also look into statistics. For example, for these 3 vegetation indices, I took plot means and also plot standard deviations of those indices. With the crop mass, I also took mean, standard deviation, and maximum values for each of those indices. And also I tested 2 different canopy cover methods, and we have 1 method for plant height. So when we combine all of this, we have 27 phenotypic variables that we can extract per flight. And once we had 19 flights, in the end, we're dealing with 513 variables. There are potential candidates for predicting maize grain yield. And what is the vision of using a plant breeding program, like all of those 513 variables? So number 1, we want to detect the phenotypic variability among the genotypes. So once we detect the variance, we do a variance analysis to look into the components of that variance. So I want to know how much of that variance is coming from a genetic component. The next step was to look into the repeatability and heritability. So how much of that variance is repeatable? How repeatable is that variable? That's what I mean. And also if that variable is useful for predicting yield within the existing genotypes and environments, and also in selecting and tested genotypes in untested environments. Okay, so here we have the first results, the results of the variance components. So instead of picking all the 27 variables, I picked 1 from each main category, and I'm going to guide you on how to interpret this data. So each plot here is a time series plot of the variance components. So here on the x-axis, we have days after sowing, so time, and here the percentage of variance. Here we have plant height, canopy cover, and the plant mean of excess green red difference vegetation index. On the variance, so this was the model we used to look into the variance. So we're trying to explain the genotype by genotype, environment, rep, range, row, and error effects. In maroon here is what we want to, what we're interested in, the genotypic effect. And there is, it's interesting that we see a temporal behavior for each of the variables. So for example, if we look here, canopy cover, we see that around the flowering time, which range from 63 to 67 days after sowing, is when we see a peak in the variance being explained by the genotypes. And we see that decreasing after flowering time, and then it will increase again by senescence, by when plants are drying out. And this is interesting because by this time is when we can capture the most of the genotypic variance is when the plants, they're flowering at different times, and also they're drying out at different moments. So this is when you can tell the most difference between the genotypes. And the same thing will happen when we look at the vegetation index. And which is interesting is that it's different in plant height, because we'd see it like increasing and then plateauing at some point, except by this outlier date. And then when plants are drying out, it starts decreasing. We can also look at, for example, environment effects, that's when we can see the most difference between the two environments that we tested, so the irrigated and the dry land. We also have some spatial error. They're captured by range and row. And also important that, spatial effects. But it's also important to look at the amount of error that we have in some days for some variables. And that gives us an insight in, so whenever there is error, there's something that is not being captured by the moment. And that's where we could look into like next steps for research, to looking into like new things that they're not being captured so far. But going back to our results, when we also look into time series and like growth curves, and here I'm plotting days after sowing as in NGRDI, so another vegetation index. Now we're comparing the growth curves of different hybrids, and then the continuous lines are hybrids that share a common parent, and the other ones, the dotted lines, are commercial checks. We see that in the beginning of the season and until they reach a vegetative peak, the lines, they kind of overlap, so the plants, they're not too different. But when they reach the vegetative peak, we see the lines scattering, and the hybrids that share a common parent, their lines, their curves overlap. So with that, we can tell that not only this this variable is heritable, but also its behavior in time is also heritable. It's kind of cool. And something that we didn't know until we look into the data is that when we look into plant height time series, again, we're seeing a sigmoidal growth curve. For plant height, plants start emerging, and then they will grow, they will plateau, and then they will dry out, and they'll be harvested at some point. But this behavior is not observed when you're looking to vegetation indices, because you will see here we have like bare ground, and then plants starting to emerge. They're becoming greener and greener. But then when tassels come out, they're not green, they're yellow and oftentimes red. And then this is why you see a decrease in the vegetation index. Except that when you have like windy conditions and the tassels will bend over, you'll be able to get more of the greenness of the leaves, so they don't have this peaks until the tassels dry out, and then you start seeing green again, and then the plants will dry out, and you see this decrease in the vegetation, the growth curve, and then it will start growing again with weeds. So this, so again, this is something that we didn't know until we see the results, the data, and brings us a challenge when trying to model like a curve for this spectral variables. Okay, so once we looked at our variables, we've seen there is genotypic variance. So what are the next steps for modeling yield? So first we want to look into the variables and look for a multicollinearity and autocorrelation. So multicollinearity, that means that more than two variables, they're correlated among themselves. And autocorrelation means that a variable is correlated to itself. And one example was with plant height. So if you measure plant height at day 50, that measure is correlated to day 40 and day 30 and day 20, because it will always build from the past measurement, unless you have lodging. But in optimal conditions, it is autocorrelated. So we also want to look into which are the most informative variables and which are the most informative flights. Here is a correlation matrix of all our variables, and they're grouped by variable, and here I'm zooming out on one little block. Here is the block of plant height, and each row within the block is one date. So when I tell you that plant height is autocorrelated, this is what I mean. So one correlation equaling to 1 is red, 0 correlation green, and -1 is blue. So one date, the measurements of one date are highly correlated to the other for plant height. And this would be a problem if we were considering like each time series as a variable. But here I'm considering each variable, each point in time as a different variable. So in the end, it will translate into multicollinearity. So why are we doing variable selection? Because if we deal with this amount of variables, there is a lot of room for overfitting, and we also want to make things simpler. We don't want to fly 19 times. We want to fly, I don't know, 5, 6 times. And the goal here was to reduce the number of variables from the model. So the model would be something like yield as a function of variable 1 at date 1 and variable 2 at date 1 until the very last one. We want to reduce it into a model of 10 and another model using 20 variables and see how well we can predict yield. And for that variable selection, I tested 3 different algorithms: random forests, LASSO, and ElasticNet. I don't know how familiar you guys are with those models, but in a simplistic manner, random forest builds binary decision trees from bootstrap samples of our variables, and then we'll randomly build decision trees, and it will select subsets at each node, and it will do permutations with those subsets. LASSO is kind of a penalized regression. It's commonly used for GWAS, and it has a shrinkage penalty hyperparameter called alpha that will shrink near zero regression coefficients to 0, and then when they shrink to 0, they're removed from the model. And ElasticNet, it's also similar to LASSO, but the difference is that in LASSO, if you have variables that are correlated, LASSO will randomly pick one and eliminate the others, while ElasticNet will group the variables that are correlated, and then we'll eliminate all at once, or we'll keep them all in the model. And then so the modeling was done in 2 steps. So number 1 was the variable selection, and number 2 was training and testing models with the variable sets selected at the first step. So we set the hyperparameters with tenfold cross-validation to eliminate that bias in LASSO of randomly picking one correlated variable. We ran each algorithm 30 times. And then I ranked the variables based on importance score. So for LASSO and ElasticNet, the coefficient of regression was the importance score, because all of the variables, they were normalized. For random forest, the importance score was the increase in the mean square error, the percentage increase in mean square error. So remember that I said LASSO would test different sets, subsets of variables, and then when you remove one variable from the model, you can observe how much of the error increased. And then the higher the increase in the error, once you remove the variable from the model, the higher is the importance of the variable. So this was used as an importance score. So all the 513 variables were ranked, and then I kept one variable set of the top 20 variables, and another one of the top 10 variables for each algorithm. So in the end, I had 6 variable sets. And I was using both environments. Next step was to keep 80% of the data set to train the model and 20% of the test, 20% of data set to test the model. And so I was using those data set to fit a ridge regression model. And also, so I tested, I've done it with the 6 variable sets and also once with the full data set, so the 513 variables. Again, I ran it 30 times, took the average results, and then I compared each model through a variance analysis. And we were looking into three parameters to tell how well the models performed. So number one is Pearson's correlation, R. The next one is root mean square error. And the third one is Jacquard's similarity index. And I found that was important because for plant breeding, it's not only important how well you can estimate the yield, but also it's important to check if you're like how well are you picking the best yielding hybrids or the best yielding genotypes. And I'll give you just an example. This is one, just look into this scatter plot. So if you plot, it's one example of one run, the actual by the predicted, and here you get your scatter plot. But still, if we make a line here, and if we're able to pick the best individuals, that would be good for plant breeding, for selecting the best genotypes. And here, a lot of information, but those are the selected variables under each of the models. So we have the top 20 for random forest, LASSO, and ElasticNet. And we also subset that into the top 10. So why did I choose 10 and 20? Because I wanted to see how well we could do with this amount of variables comparing to using the 513. Something interesting to note is that from the 19 flights with random forest, we could go down to 6 flights. With LASSO, 9 flights if we're looking at the top 10, and 11 if we're looking into the top 20, and 8 flights if we look into the ElasticNet selected variables. We had variables from all the categories being selected. And something interesting is that plant height was often selected at late dates, but towards the end of the season. And some variables were common in all 3 models. For example, canopy cover at 60 days after sowing was the top one for the three models. Canopy cover at 105 days after sowing was also in the 3 models. Canopy cover again at late in the season was in 3 models, and some were present in 2 models. So for example, plant height was in the LASSO and ElasticNet, and also the NGR the plant and giardia, I mean, was also in this too. And how does this the model? Yeah, how do the models compare? So when we used all variables, the correlation to yield was 0.67, and the lowest value was 0.61 using random forest top 10. So again, here we have LASSO top 20 variables, LASSO top 10, ElasticNet top 20, random forest top 20, ElasticNet top 10, and random forest top 10. The models with the same letter, they are equivalent. They're not statistically different. So that's very encouraging. So for example, if we look into the top 10 variables selected with LASSO, that is similar to using all the variables. Another interesting thing is that there is a difference when we look into the different environments. So here we have all the 7 variable sets, and then the results of Pearson's correlation for each environment. So here on my room, we have all environments. On green, the dry land, and on blue, we have the irrigated. So we can actually have better results with the irrigated environment. So if your environment is stressed, so you have this water stress, the models have a little bit of a hard time in correlating your variables to yield. So when we look into the root mean square error, so here we're talking about tons per hectare. Here it's the opposite. Lower values are better, so we want to have the lowest error values. And again, LASSO 20, LASSO top 10, and ElasticNet top 20 variables were comparable to using the full data set, and the mean of the root mean squares errors that we got was 1.08 tons per hectare for all variables until 1.16 using random forest. And again, we see a difference between the environments, with the dry land having higher root mean square errors. Once we look into the similarity index, we have 0.3, 0.32 if we use all variables. And the least value is, again, for random forest top 10, 0.23. But these models are comparable with using a model with all the variables. And the same thing happens, the same difference between environments is observed. So what can we conclude from this study? So yeah, that is a genotypic value and an yield prediction potential from those variables extracted in this study. The flights with highest variance explained by the genotype occur during the flowering time and also in senescence time. Even though, like for example, when we're looking into correlations and similarity index, even though those are not like the best values, there's still a significant portion of the variance like being explained by the genotypes, and also the variables are explaining a significant portion of the yield. Because I would say like as a matter of use, it would be interesting to prioritize early flights. So we saw that during flowering and also during senescence are the two like most important dates. But if we could prioritize early flights, that would be beneficial for plant breeders to make their decisions in advance and also plan like for future trials before harvesting. And we also see, like, if you think of, oh, how would you apply this model to different years, different genotypes, because we were dealing with specific points in time. So another way to make this model more robust was by considering each variable as a time series and not each point in time as a variable. And with that, you could just like plot a curve, and then you could like interpolate the data from whenever you fly. And next steps would to like perform tests in predicting known genotypes in known environments, unknown genotypes in known environments, known genotypes in unknown environments, and finally, unknown genotypes in unknown environments. With that, I'd like to thank my committee members, my chair, Dr. Seth Murray, and then sorry, Popescu, Dr. Murillo, Maeda, Dr. Jinha Young, Dr. Anthony Filippi, all the students, the grad students and student workers, and Dr. Murray group. Also, everyone involved in the UAS team and other G2F collaborators, Monsanto for the fellowship. And thank you all. And with that, I'll take any questions.
Thank you, Nathalia.
No problem.
We would like to ask that the first question be from a student.
Mm-hmm. Let me see.
The speaker will be over here. Hi, Nathalia, can you hear me? Hello.
Hey.
Can you hear me?
I can hear you. It's kind of low, but let me see if I can understand.
I have a question here.
Okay.
You were comparing 3 different models, and on your variable selection, you found different amount of flights to better explain your models based on each model, right? First one was 6, the other one was 8, and I think the other one was 9.
Yeah.
So my question is, were those flights the same?
Good question. So there were a lot of the flights that would be the same. The same thing. I can actually. Yeah, I didn't have like a number to tell you like the similarity, but a lot of the flights we could find them on the same. So for example, 60 days after sowing would appear here, like on 3 of them. So 105 would also appear in all of them. So there were a few flights that would be that would happen in all 3 scenarios.
Okay, so among those, which would you suggest for a breeder to start flying to better collect and predict whatever they want to predict?
Right. So so again, the most informative flights were the ones that happen around flowering time. So especially like when the flowering started. So this is where we could capture the most difference between the genotypes. Around senescence, they were also informative, but I think that would be like too late for like getting like useful information as you're so close to harvesting. And also from my experience, if you want to like this is something that we didn't look into yet, but something cool to see would to look at, for example, the the speed to which the plants like develop and cover the ground, for example. But so far, with the information that I have right now, would be flights around flowering time, or just in a little bit just before the start of flowering.
All right, thank you.
No
Good presentation.
problem.
Very much.
Thank you. Mm-hmm.
I have a question. Could you hear me?
Yes.
The reason I think you might use the UAV is because it's accessible and convenient. But giving it the performers or whoever want to apply this kind of prediction to fly like weekly and then bi-weekly, but that's can also be a burden. So would it be possible to switch the platform from UAV to probably like some other already existing sensor, like hyperspectral data, or even high resolution remote sensing imagery that they can just download from the like USGS like website would be better to do that?
Very interesting question, and there are a lot of points to consider in that. So the best choice of your remote sensing platform will depend on what are you trying to do. And so, for example, if you want to do plant breeding, you will need something with like very high resolution. And so for plant breeding, UAVs are able to give you that resolution. If you're looking into, because you're looking into the plot level, but if you're a farmer and you have like you're doing extensive planting, so you don't need to have this 2-centimeter resolution. So with that, depending on the size of your farm, you'll be able to acquire image from satellites. I think there's a Sentinel satellite that has, I may be wrong, but I think it's like 10-meter resolution, and that would be enough for a farmer, for example. And Sentinel, I think it's free. You have free access to the images. At the same time, so again, it depends on what you're doing. We flew every week because we wanted to know, we want to do this job for the plant breeders. So we flew every week so they don't have to. So we want to look into the time series and pick the best, be able to pick the best moments to go to the field. And does that answer your question?
Yeah, thanks.
Okay, no problem.
I guess you explained it really well.
Yeah.
This is, go ahead, Dave.
Okay, can you hear me all right?
Yeah.
Okay, so nice seminar, really interesting. I'm kind of curious, so given your set of selected variables, are quite a few of those correlated with each other? And if so, how would that, how would you process that information with regards to retention or revision of your list of selected variables?
That's very interesting question. So I would say that there is, yes, there are variables that are correlated. So for example, the vegetation indices, they should be correlated because they share the same bands. We use the same bands to create them. I would expect like plot vegetation index be kind of correlated to canopy cover and plant vegetation index because it contains information that are contained that exists in both. So how we would select among them. So I would pick the one like the ones that they're most frequent in my selection ranks. And I would also say that the advantage of using one or other variable is that there isn't like a monetary cost to them. Like there is a cost in flying more, but there isn't this cost in adding one or other variable. So they would be like a computational cost, but that I think that wouldn't be like a big deal if you want to use a bigger data set in variables. I hope that answered your question.
Yeah,
Mhm.
yeah, it does, and yeah, you have to think of it in terms of
Yeah.
you know, what causes extra work.
Yeah.
And so flying on different dates or whatever, and maybe the setup cost for your scanning system would be important. So yeah, it's quite. The other thing, of course, you were just looking at yield, right? So
yeah,
I mean, is that going to be the extent of it? Is looking only at yield, or do you envision additional factors coming into play, goals coming into play?
So yeah, for my work, I'm looking into yield, but there is an immense potential in those variables to look at different aspects of the crop. So I could look into nutrition or disease or pests.
Drought susceptibility and so on. Yeah. So, okay, great. Well, thank you.
Okay, thank you.
Natalia?
Yes.
Fernando speaking. Very nice presentation.
Thank you, Fernando.
I have a couple of quick questions. In your results, you have shown that across the multiple times that you collected data, it was clear that the highest genotypic variability was detected at 2 instances, right? One was, I think, flowering, and the other one was late at maturity, I believe. I was wondering if with all this data that you have collected, you didn't concentrate in only these 2 instances and trying to find those traits with that would allow to maximize the yield of prediction only within those 2 instances. Did you try that?
I didn't try that, but that is a great idea.
But I mean, the part of the goal here is that we'd be able to make selections earlier in the season. So if you're finding stuff at the end of the season, it's not really all that helpful. So in some of her models, she is identifying things. I mean, you can see things at 60 days after planting, and that's sort of right before flowering, or actually a ways before flowering. So that's really helpful. I mean, that's much more helpful than finding something the day before you run the combine through the field.
Yeah, I thought that 60 days was close to flowering though, right?
Yeah.
Okay. The other question that I have for you, Nathalia, is, you know, in plant breeding, it's even today a very hot topic, trying to find efficient strategies to do selection under dryland conditions, especially in corn. And even today, there's a decent amount of effort that is trying to devote to do selections under optimum irrigated conditions for dryland conditions. It's a form of indirect selection. With this type of technology, do you think that's feasible based on what you have seen?
Could you repeat your question? I think I got lost at some point.
Sure. The essence of the question is that you assess this study under fully irrigated and dryland conditions, right?
Yeah.
My question is, do you think you can make some form of predictions based on your results under fully irrigated conditions for dryland conditions?
Oh, I see. I think It depends on how you train your model, because like we have like data set for one season, right? I think you have enough like data in your training set that would comprise both like dryland and irrigated. Like why not? But I think like so far, like with this data set, it was like more of a proof of concept than like building a model that will be like really robust and predictive or of other scenarios.
Okay. Thank you, Nathalia.
No problem.
Hi, Nathalia. Very much appreciate your seminar, and especially the leaning towards and the benefit for plant breeding programs, how to best optimize what we're doing. We've been flying a lot in the wheat breeding program. Amir Ibrahim and Shannon Baker here in Amarillo. The question is, we've been using RGB and multispectral. You used only RGB, but yet your vegetative indices seemed actually pretty good. Do you have any comparison between NDVI and what you got through RGB?
So we don't, and the limitation was like in the data set, because like when you're flying multispectral, you have to have like radiometric calibration and all that. So we could not calibrate it to make it consistent throughout the dates. And then we still wanted to like push it forward with the data set that we had. So this is why we only use the RGB, and we're not comparing.
Okay, I understand that. We've just been doing exploratory for the last 3 years, and we're ready to kind of reduce our workload, so to speak. And have already considered that RGB by itself really would provide a fair amount of data, and that would cut our workload in half. Right now we fly RGB, we land the drone, get another one to set up and fly it with multispectral. So just kind of thinking out loud here.
No problem. Yeah, it makes sense. Like why make it complicated when you can make it simpler, right?
Jackie, our group did get the new Phantom 4 this year, which really wasn't all that expensive, which adds a red edge. So Scott's flying it now, and it's one flight and you get both. So that reduced it quite a bit. But the other thing to keep in mind here is that, and Nathalia said this, is this is proof of concept. So what I sort of challenged her with is, how can we get as many variables as possible? So as we're moving from this idea of, single traits like measuring plant height into predicting yield, we also have to have the mentality not of QTL mapping of single loci, but of genomic selection, right? And having as many different random variables throughout the genome, or having as many different random phenotypic variables as possible. And so based on the algorithms that she developed for these various vegetation indices, these are the top candidates. But we can conceive that in the future, as more extraction algorithms are developed for different, I don't want to call them traits because they're not traits, but plant height, NGRDI, but then there's plant texture, or maybe a leaf angle or solar angle on the leaf. You can imagine extracting thousands of different features that could then be put into a predictive model. And so I would be hesitant to get rid of anything and say right now these are the most important, because I think this is really just sort of a proof of concept at this stage.
I understand. Thanks, Steph.
We are right at 1 o'clock. So I want to thank Nathalia again for your presentation and for being part of the Plant Breeding Circle. And I'm going to go ahead and close down the recording.